on migration and a few things related towards that. And then we move on to taxonomy and systematics and ultimately ecology. So the first part of this is how fishes sense their environment. So a couple things to recognize. The obvious thing is that fishes live in water. Water is a very different medium than air. Water is about 800 times denser than air, and it can't be compressed, which means vibrations such as sound travel through water very, very, very well, right? So basically sound travels through water about 5 times faster than it moves through air, okay? So that's what it's real easy for us to pick out directionality of sound, because it's slow enough that the timing difference between, like say sound coming from over here hits my right ear first and then my left ear. And that's enough of a timing difference to tell that the sound's coming from over there or from over there or wherever, right? But for those of you that have spent some time underwater and you try to listen to sounds and figure out where it's coming from, you can't tell because it's so fast that you can hear, you know, their sound, but you can't tell where it's coming from, right? So like divers, you're coming up towards the surface, you hear boats around, but you can't tell which direction the boat is coming from, and so on. So that becomes a thing for fishing because they have to deal with that type of environment. And so ultimately, think about sound, it is vibration moving through a medium. In this case, the medium is water. So fishes have a structure that's sort of the basis for all kinds of vibration detection that is known as sensory hair cells, okay? This is sort of a typical sensory hair cell right here. So there's a neuron that's going to ultimately connect into the nervous system. But for our purposes, what we're concerned about over here is the kinocilium and the stereocilia, okay? The kinocilium is this structure right here, and it is basically fixed. It is a rigid structure. The stereocilia, these little sort of finger-like structures here, are flexible, okay? So basically, as you get vibration, the stereocilia are going to either move closer to the kinocilia or away from the kinocilia. And that then affects, or that determines, I don't want to say this, whether the fingers, whether the stereocilia are getting closer or farther away, gives the fish that sense of directionality, okay? So that's how they can determine which way the sound or the vibration is coming from, okay? It doesn't matter to us whether they're getting closer or farther away to determine like whether the sound is getting closer or farther away. It's simply that difference between the two that gives the fish their sense of directionality.
Now, wouldn't the vibrations or sounds like travel through the otolith structures?
We're not at otoliths yet.
Okay.
We're getting there. But this is, so these are involved, but these aren't part of the otoliths, right?
Okay.
But otoliths can vibrate on top of these where otoliths are present. But this still occurs even if otoliths are not present, right? So these are just, so we're starting with our base structure that picks up vibrations. And it's simply the relationship between the stereocilia and the kinocilia, okay? So that's how they detect vibrations. Okay, good so far? Anything else? All right, so there are numerous types of structures that use these sensory hair cells. One of these structures is called a neuromast. I think I have it on the next slide, but that is this structure right here. So the neuromast is a collection of hair cells. It's a collection of these sensory hair cells. So that's what each of these little structures is right here, are the individual structure sensory hair cells. Okay? The stereocilia and kinocilia of all of these hair cells are all sort of gathered together under this structure that's called a cupula. Okay? So basically it's just a cup that goes over the cilia. Okay? So now what you have is this slightly larger structure that will pick up the motion of the water and therefore trigger the sensory ability of the stereocilia relative to the kinocilia. Okay? So these neuromasts are an important way in which fishes in their in the lateral line system can pick up movement of the water. Okay? To get a sense of how sensitive their system is for this, you go stand in a creek, right? So take your shoes off, leave your shoes on, I don't care, but stand in there with shorts on or something, and you can tell which direction the water is moving. But that is it, right? But you can have the fish that is in that same stream, and picture like in the riffles with fast-moving water of an Ozark stream. You can feel that water move. The fishes that are in that water can still detect other fishes moving, right? So that's how sensitive this system is. And in part, it is because of this extensive lateralis system that's driven by a whole bunch of sensory hair cells combined together into a whole bunch of neuromasts. Okay? Are we good so far? All right, so the neuromast can be present either inside of a canal. The lateral line system can form a canal. Remember when we first started talking about scales, we said some of the scales of the lateral line system will have pores, right? So those pores are an opening through the scales that allows the flow of water to go into the lateral line canal, into the lateralis system, okay? So you've got these scales, and that's what sort of this darker gray right here is that canal system. That's our lateral line system. And the individual black dots are indicating the pores, okay? So water can get in and it flows generally in one direction through the canal. So that flow triggers the movement of the cupula on the individual neuromasts, right? And there will be several neuromasts scattered throughout the canal, okay? So all told, you have this nice sensitive system for detecting flow of water. All right, so those neuromasts that are down inside of the lateralis system, down inside of the lateral line, those are known as the canal neuromasts. Okay? But some species of fishes also have superficial neuromasts. And superficial means that there are individual neuromasts just directly on the skin of the fish, right? So they are not inside of a canal, they're on the skin. Fishes that tend to live in areas with lots of fast-moving water typically have lots of lateral line neuromasts, but no superficial neuromasts, right? Because they're directly exposed, they're not inside of that protected canal, they're directly exposed, fast-moving water just drive them crazy, right? But if you are a species that lives in primarily quiet water, then having superficial neuromasts on the skin increases your sensitivity to movement in quiet water, okay? Now you can still have fishes that live in quiet water that still have canal neuromasts inside of a lateral line. So really all we're talking about here is sort of the relative abundance of the superficial neuromasts. Quiet water tend to have more species with more superficial neuromasts, but in moving water tend not to have the superficial neuromass. Okay? Any questions? All right, so because the lateralis system is designed to pick up motion in the water, to detect those vibrations, any vibrations produced by the fish itself could end up triggering sort of some false responses, right? So notice many species will actually have adjustments to the lateralis system. It's not a straight line, but instead you might have an arch or just sort of a change in the placement to go up and over the pectoral fin, right? And that way, movement of the pectoral fin, right? So picture a fish that's just sitting there, they're often just sort of beating their pectoral fins back and forth, right? So that creates motion in the water. If you have the lateralis system right below it, you're going to pick up on a lot of that. And it's going to sort of block your ability to pick up surrounding motion. So instead, what you have in the lateralis system, the lateral line goes up and around or is above it, and that way it's not picking up the direct vibrations from the movement of the pectoral fin. You got a species that is a bottom-oriented predator, right? They're hunting above. Their lateralis system is towards the top of the body in many cases, which makes sense because their prey is up here, right? Got surface-oriented fishes, such as a flying fish. The surface of the water is above them, so their prey aren't really there. Their prey are below them, so their lateralis system tends to be farther down below. Now we can get to otoliths.
I knew I was foreshadowing at some point.
You're always setting me up for some point in the future. Sometimes later that lecture, sometimes 4 lectures down the road, but you're always setting me up. All right, so one of the things to bear in mind, we talk about the density of water and it transmits sound really quickly. The body of the fish, the overall body of the fish, the musculature and so on, that density is more or less the same as the water. So what that means is that sound that is traveling through the water or vibrations, they're traveling through the water, right? They pass through the water and they will basically pass right through the body of the fish and the fish won't feel it. They won't notice it, right? But there are structures called otoliths. They're sometimes called ear bones or ear stones. They're calcium carbonate or bony type of structures that are denser than water. And so what happens then is when the vibration hits that otolith, it causes the otolith to move. Right? So the fish won't feel most of that sound pressure, but it hits the otolith and it causes the otolith to shift position. There are 3 different otoliths, okay? There's actually 2 of each one. These are paired structures. They're located in the inner ear, right? So we have inner ear canals, which we use are important for our balance, right? So they have the same type of structure, and I got some really nice photos to show you in a second, but have the same type of structure in the inner ears, and they have a really large sagittal that is by far the biggest of the otoliths. The one that I had out the other day in the tray over there, that was one of the sagittal otoliths, okay? But then there's also a lapillus and a stericus. Now lapillus is primarily used for balance. I'll talk more about that in just a second, okay? Whereas the other 2, the sagitta and the astericus, are typically used for sound, okay? Now, whether it is for balance or whether it is for sound, the overall interaction of the otoliths and the sensory hair cells is basically the same, okay? So essentially what you have is a bed of hair cells, right? So you got all those hair cells up there and the otolith rests on top. So if you have that sound that's passing through and it causes the sagittal otolith to move, that movement triggers vibrations in the hair cells, right? So the hair cells pick up the vibration, which they can translate as sound. Same thing for the astericus, okay? In the lapillus, it's essentially gravity. So you have the weight of the lapillus otolith pressing down on the hair cells, right? And so that gravity pulls down. Use that in combination with eyesight, and this fish has a sense of, is it upright or is it turned over a little bit, right? So they get their sense of balance. So it's the weight of this pushing down, where the other 2, it's the vibration that gets translated as sound vibration. All right, we good so far? Now, we can also think about sound relative to the skin body. We said the body of the fish is about the same density of water. So that sound vibration just moves right on through the body of the fish. It's a dense otolith, causes it to move. But at the same time, the swim bladder is a pocket of gas, right? It's basically a pocket of air, which is much less denser than water, which also then can pick up or detect the vibrations that are moving through the water, okay? So sometimes the swim bladder can actually be used to help sort of amplify the sound. There's a group of fishes known as the odophysants, okay? You'll learn more about what those are, but that includes like minnows and shad and things like that, that actually have a bony connection. So here's the swim bladder, right? So that's that big bubble of air, right? And you've got this, what are called Weberian ossicles. Weberian ossicles are bony. Like there's just little bony modifications. They've evolved from the first 4 vertebrae of the backbone. But they have these structures that actually come in contact with the swim bladder and then ultimately come in contact with the inner ear. So any sound that is picked up by the swim bladder has a direct connection and sends that sound to the inner ear. Right? So that's another way of fishes being able to pick up relatively faint sounds in the water, right? Relatively faint vibrations, because that swim bladder is like a big old echo chamber. All right, questions? All right, so just a couple of pictures. This is looking down on, so here's the dorsal surface here, you're looking down on the fish. So this is the brain, this is going towards the front end of the head, the anterior end. So you can see the really delicate inner ear canals, and then you can see some of the different otoliths. So here's the side view. So again, here's dorsal, here's the head of the fish going this way. So there's the astericus, there's the smaller lapillus, or lapillus, however, and then the big old sagittal bone. So that's always the biggest one. Again, remember these are paired. You've got one on each side, just like we do. So essentially all vertebrates have this structure. We don't have these big old bones, right? We got tiny little particles, but still the same thing. Basically tiny little bony particles that are still present over sensory hair cells. So we still use them for partly sound and for balance.
Okay, you say it's a really big bone, but how small are we talking to? Like
That's
scale?
relative to the size of the fish.
Okay.
Right? So the one I had out here the other day, what was that? Like the size of a dime or so? That was, it was from a fish, it was probably from an Asian carp that I found along the Mississippi River, right? Bigger fish will have bigger, some of the sagitta can be like that long.
Okay.
Most of them are going to be like a dime or something like that for a larger fish. Way back in the day when I was doing work with gobies, they were like split pea size, right?
So you about needed a microscope to see.
Oh, I had to use a microscope, yeah. Because the other thing you can do, some of you more fisheries oriented maybe already know this, but these things grow just like trees do. So they lay down rings. And so you can actually age the fish. So you cut it basically in cross section. Typically you'll treat it because you have sort of a winter time and a summertime, and so you have a softer tissue between harder tissue, and so you can etch it and it'll create tiny little ridges, and then you can look at it either on, usually they'll use like a electron microscope or some other way of being able to then count the number of rings that are present and so on. Larval fishes put down one ring every day, and so you can actually count at the rings. And there's a sort of what's called a settlement check, sort of a distinctive mark from when they transform out the larval stage into the adult stage. So you can essentially count from the center out to the settlement check, and you can determine how long that individual was in the larval stage for. So, and then the other side, just a different view, but again, the same thing. So I just, as soon as I saw those pictures, like, I got posted because they're just cool. All right, chemoreception, smell. So chemical cues do transmit through the water. right? The same way that chemical cues go through air, right? We pick up those odor particles. Those odor particles can move through water as well. So fishes have this large olfactory structure. It's sort of what it looks like right here. So essentially you have two pores, right? And if you look on the side of a fish, you'll usually see two little openings, right? The nostrils. And so you have an in current there. That's basically where the water goes in. Water flows across the olfactory gland and then goes out the other pore, right? Again, you'll have that paired system on either side of the head. So you've got two of these, and then that is picking up the chemical cues, okay? So basically the same way we pick up things, right? We smell, and then we got chemosensory glands in our noses, and we pick those up. Now, the interesting thing, we think about our taste buds present in our mouth, primarily on our tongue and so on. Species of fishes can have taste buds all over their body. That's kind of a weird thing, sort of tasting the world around you. So a lot of them, of course, are in the mouth, but they can be on the lips, they can be present on barbels. So, for example, in this goatfish right here, they will cruise along the bottom, and their barbels are working the bottom, and they're basically tasting the bottom. So they're picking up chemical cues that are present from potential prey items that are buried down in the sediment, right? So, but they can have it on their skin, their fins, and so on. There are a group of fishes that can produce what is sometimes called an alarm substance. It's called Schreckstoff, and that's because it was originally discovered by a German who came up with this. Schreckstoff means alarm substance, right? Or basically alarm stuff, right? But that's essentially what that is. So just picture Shrek. If you saw him, you would be alarmed, right? So, or you'd be like, dude. So, you'd be like, okay, donkey. I watched Shrek in a long time. So anyway, but Schreckstoff is a substance that is present in the skin of some groups of fishes. Okay? Again, they're in the, what is right up there, the Asteriophyzons, which includes the Odophysons. Again, I know you can't wait to get there, but you will learn that. But what that does, let's say that you've got a minnow. So a minnow is one of these species that can produce Schreckstoff. Okay? So what can happen is that, let's say a predator bites one of those. It releases this Schreckstoff into the water, and the other fishes pick up on it, right? And then they can respond accordingly. So that's what this figure right here is showing. So it's kind of hard to tell, but the little black line is the before they put the stuff into the water, and then up here the red is going to be the response after they introduce the Schreckstoff into the water. So darting behavior, right? Suddenly jerking away to get away from a potential predator, right? Basically didn't happen. But as soon as they put in this Schreckstoff, right, they basically extracted it from the skin, released it into the water, notice that the amount of darting increased dramatically, right? So the idea is you can dart away from a potential predator. There's something called slow swimming, which I really don't know. It's a known alarm behavior, but I don't know why it's called slow swimming. Often they'll do darting and then they'll sort of slow down afterwards, but I don't know if it's like slow directional, kind of away from where that is. But whatever it is, it increased, right? So again, basically wasn't present, but once this Schreckstoff was released into the water, then this behavior, sort of the anti-predatory behavior, also increased. Now this was done, and this study came from zebrafishes, really common aquarium fishes, right? Go to the pet store, you'll see the little zebradanios and so on right in there. They tend to form in larger groups near the surface, okay? But when this Schreckstoff was released into the water, then they went down to the bottom of the tank. And that's what this figure's showing. And I really set that up because it's weird, because this is the number or proportion of individuals that went down to the bottom, but the bar is actually higher because it's a percentage, right? So a high percentage went down to the bottom. So to me, it's always been a weird figure to look at, because you're like, no, they went up. But it's like, no, they went down, right? But that's the idea, is normally most of them are up near the surface. But once the Schreckstoff was there, Basically, they dart more, and they move down to the bottom of the tank in this case, right? So again, predator avoidance.
Wouldn't that make you more of a target if you swim to where you can't escape?
They're in an aquarium, they don't know, right? But
Right.
but you, if you are, think about them in the wild, right? If you're normally up near the surface, this is where the predator was, right? Because the Schreckstoff is there because the predator bit a fellow individual, right? And so that releases the Schreckstoff. So if you're up there and the attack was up here, what are you going to do? Go down, because then you get away from them.
But marine biology wise, don't they usually start from the lower and then go up
No.
to the top to hunt?
No, not necessarily.
I
It all
remember
depends
that
on
wrong,
the,
then.
it depends on the species of fish, right? So in this case, this particular species, bear this in mind, for this particular species tends to live near the surface or just hang out near the surface of the water, right? So, and that's why a lot of minnows do that. Not all, but a lot of them do that. So if there's a predator up there, then it is to your advantage to go down deeper because the predator was here, so you go away from the predator. The darting response also sort of gets you away from the area where that's concentrated, but it's basically go away and down. Anything else? All right. Some species of fishes, a pretty fair amount, have the ability to pick up electrical signals. All living organisms, all living animals, most living animals, slowly backing off here, they have a central nervous system. They're producing electrical signals, right? Because that is how that's how the nervous system works. It's a series of nervous impulses, right? Or electrical impulses. So some organisms have evolved the ability to detect electrical signals. Some have evolved abilities to use that electricity in various ways, okay? There are two types of structures that pick up electrical signals, okay? Ampullary receptors pick up electrical signals, weak electrical signals, passively, right? So basically they're just sitting there and they pick up any electrical impulses that happen to be passing by. So this is really good. It's really common in sharks and rays, but a lot of other species have them too. Sturgeons have them. So all of these little white dots you see all over right here, those are ampullary receptors. So sharks will use them, for example. So this is a chimera. They'll often feed down near the bottom, and they can pick up electrical signals emitted by animals that are buried down in the sediment, right? So then they can go and attack them. Same thing, so you have fishes that are swimming around, they're producing electrical signals. Sharks with their ampullary receptors can pick up on them and hone in on the prey, okay? But other fishes actually have tuberous receptors, which they use as part of an overall electrical communication system. So they are purposefully generating electrical signals and purposefully detecting those signals. What they're basically doing is generating an electrical field around themselves. Okay? So they generate an electrical field, and if a prey item moves into that field, it distorts the electrical field, which then the fish can detect, right? So they have picked up on the presence of a prey item. So this is really common in some of the ghost and knife fishes, the mormyrids. You'll see a picture of one of those in a second, right? But they are producing they're producing electrical fields that can be distorted by prey. But here's the other thing that's actually kind of cool about this, is that each species has its own species-specific electrical signal. So they can determine individuals of the same species or of different species, right? So they can say, you're like me, you're not like me. So for example, this right here, you've got this species, you've got this species, right? So this is essentially the waveform, the frequency. So notice that each time you go up or down, you basically have a pulse. It'd be like a high frequency electrical signal right here. This one, notice basically a weaker pulse, a pause, a stronger pulse, a pause, a weaker pulse, etc., right? Very different patterns. So looking across this relatively diverse group, each species had its own distinctive electrical signal, right? So that can be important for within species communication or within species recognition. Now, no two individuals of the same species produce exactly the same frequency pattern, okay? So right here, you got two individuals of the same species. Hertz is frequency, right? So 500 hertz, relatively low frequency. I practiced this today. Terrible whistle, but that is roughly 2,500 hertz, right? So 5 times lower frequency, that's getting down into the 500 hertz range, okay? So that's fairly low frequency. But here's the thing. These two are similar, but they're not exactly the same. If you have two individuals producing slightly different frequencies, those frequencies interfere with each other, and it inhibits each individual's ability to detect what's going on around them. So what happens is when they occur together, in this case, the species that has a slightly higher frequency actually shifts to an even higher frequency. The one with somewhat lower frequency shifts to an even lower frequency. So there's a greater gap between the two. So instead of having an interference, you get a nice smooth signal that they can both work with. Right? So that's what you actually can have this. It's really important, but again, it'd be just like if we were all talking in more or less the same frequency all at the same time, couldn't hear shit, right? It just sounded like noise. But if we start to separate out, some people talk higher, some people talk lower, then you can start to hear individual conversations. And that's essentially what this is doing too. By shifting the frequencies out, it becomes easier for them to coexist and still be able to detect their surrounding environments.
So would you maybe have
I
the
personally
same frequency in this part of the ocean, and then you could have the same frequency in this part of the ocean over here, and they wouldn't change them both because it's like
right, if they're far
different-
enough apart, and I don't know what the distance is, but if they're far enough apart, it doesn't matter that they have similar frequencies, right? So just again, if you and I were similar range of voice and you were talking out over there in the hall and I'm in here, we're not affecting each other's conversation. It's when we're close to each other that occurs, and that's where you then get the frequency shift to make it easier. Anything else? So some fishes can generate a whole bunch of electricity. So for example, torpedo rays, and there's a small one in that blue tub, so that'll be one of the species you have to know. I don't know how well you can see this, so you've got the overall wing, the modified pectoral fin right there. But if you can sort of see that little kidney bean sort of shape right in there, sort of same, so right in there. Again, when you get that out, take a look for that, because that generates electricity. So torpedo rays and some others, right, some of the electric eels and so on, have the ability to generate massive amounts of electricity that they use to stun their prey. And if you happen to pick one up, you're seining, you go through the beach, you're seining, and you pick one up with your bare hands and you're standing there in the water, it will be quite the zap. All right, we good? Not much to say about the eye. Modifications to the eye and the lens for focusing underwater, right? If you've opened your eyes and looked underwater, it's very blurry for you, but for the fishes, it's pretty clear. Some species, so sharks, for example, sturgeons, they have a structure at the back of the eye that's called a tapetum lucidum, okay? It's guanine crystals. But guanine actually tends to help reflect light that enters the eye. And so if you get like a shark and you sort of go like this, or if you got a live sturgeon, you go like that a little bit, you'll see that shiny eye, right? If you've got cats, or you may be seeing the lights reflect out of the eyes of a deer or raccoon, some of the nocturnal animals, that type of thing, you get that really big shine that comes out of their eye. That's because they have that same structure, the tapetum lucidum, at the back of their eye. So fishes that are active at night, or down deeper where there's just not as much light available. Having that, you get a little bit of light, then that tapetum lucidum reflects it around inside your eye, which basically increases the effectiveness for the light that is present, so you're able to see better. Now the last thing here is that all eyes, whether ours or fishes, for vision, it's a really high oxygen demand, right? Basically, it's a structure that takes a whole lot of metabolic energy, which takes oxygen, right? That's why fishes that live down deep or in caves tend to lose their eyes pretty quickly, because there's a high energy demand for eyes. But if you're in a place where there's no light or very little light, then why produce the structure, right? So there's a high oxygen demand. The retina itself is not actually vascularized, right? So it doesn't have blood vessels. So what do you think might be a suitable physiological process that allows you to build up high concentrations of oxygen in the eyeball? And you know the answer. Counter-current exchange, right? So in the choroid gland behind the retina, you have a mirabilis, right? Which again, you've got a network of capillaries where the blood flows in opposite directions, and you get counter-current exchange that allows you to build up really high levels of oxygen to meet the oxygen demand, right? Told you when you learn counter-current exchange, just get that down. And it is useful in so many different ways, right? Birds that stand on ice all the time use counter-current exchange to keep heat in their body, right? That type of thing. All right. So, you've got eyeballs to see the world. One of the ways in which you can communicate visually is through the use of color. This is not unique to fishes. Lots of animals use color in some form for communicating, right? So, no difference for fishes. So I'm not going to really focus on it too much. But one of the things that's important, again, this is important even for many land animals where color can be an important part of, like, say, their mating process, right, or mate detection. Because if you have bright colors to attract a mate, the problem is that those bright colors also make you highly visible, or certainly more visible, to predators. Right? So now you got a trade-off, because this guy's out cruising for a date on a Saturday night, but the predator out there is hungry, right? So you're highly visible. And studies have shown that in a lot of these different species, females preferentially choose the more brightly colored males, right? But what that does is that also increases their vulnerability to predation. So males that tend to be present in places where there's an abundance of predators tend to be drabber, right? You're not as brightly colored, but you're alive. Or certainly a better chance of being alive, which still increases your chance of a date on Saturday night, as opposed to the brightly colored individual that maybe doesn't make it to Saturday night. And then finally, if you're underwater and color is important, that often implies relatively clear water, right? So that other individuals can see that color, such as the difference between males and females, right? So for many of the cichlids over in the Rift Valley lakes over in East Central Africa, for example, right? Any one lake, they're huge lakes, but any one lake can have over 500 species or more of cichlids, depending on who you talk to. Lots of diversity. And for some of it, the female is choosing among the different species, the males of the different species, based on color patterns, right? What's happening, though, is that a lot of these lakes are becoming highly polluted. And so instead of having relatively clear water, it's murky water. So with murky water, the females can't distinguish males of her species versus males of other species, and she will just mate with anyone, right? So having that color is really important, But as the environment changes, they're losing the ability to detect that signal, which is then affecting increasing rates of hybridization and so on. Should we update now? I don't think so. All right, any questions? Go to coral reef fishes. The two most common colors that you find, definitely not all of them, but the two most common colors you find are blues and yellows. Okay? And so we look at these lovely fishes and we say, oh man, I can see them really easy, right? They pick out. But for predators underwater, as it turns out, those colors actually create a disruptive camouflage pattern. Okay? And the way to think about this, think about what they call the space light, the average color of the background. Okay? If you were to sort of be back and sort of out of focus of an overall coral reef, the average color tends to be towards the yellow end of the spectrum. If you're off the reef and in the water, the average color is blue. Right? So if you are a fish like this royal grama right here, then if you are against the reef, part of your body tends to blend in, so you don't look like a fish. If you're out in the water, part of your body blends in, and you don't look like a fish. Right? So you see this sort of common blues and yellows, swirly patterns, half and half types of things. But again, as the fish moves around relative to the position of the predator, oftentimes part of the body is going to blend in with the background, regardless of whether they're up in the water or down closer to the reef.
This may seem like a stupid question, but could some fishes be potentially colorblind?
Me if I was a fish.
So the short answer, purposefully yes, in other cases no. Okay, or I don't know. Let me rephrase it.
Well, in this case.
Well, okay, so let me get to the point here. So some of this you can have just like any individual. Mm-hmm. There is the possibility that their color detection system is basically the same as ours, okay? And the reason we have the most common form of color blindness is red-green, right? And it's because the red and green opsins are right next to each other, and genetically, they are really, 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 really similar. And so what can happen is during crossing over, you can get mismatched, and it creates basically a non-functioning red opsin. So you don't see the red end of the spectrum, right? And so red-green color blindness, you can see green, but anything that is red looks green. Mm-hmm. Okay, so that's the most common color form, or most common form of color blindness. There is nothing to say because the arrangement of those opsins on the chromosomes is the same. They're right next to each other. Mm-hmm. So potentially that could happen, okay? Mm-hmm. But nobody goes around and studies fishes and says, are you red-green colorblind? They really don't answer the questionnaires. They don't do that very well. So, but let's take it in a different direction, okay? It's shallow water. You basically have the full end of the color spectrum that we can see, right? So basically, let's just say blues through reds. So, shallow water fishes tend to produce all three opsins. So there is what's called a blue, a yellow, and a red opsin. Okay? Opsins are the proteins that pick up the specific wavelengths of light, right? So you pick up the blue end, the yellow end, the red end, and so between all three, we can see all 16 point whatever million colors that the human eye can detect, okay? In shallow water, all three colors penetrate to some extent. And so shallow water species tend to produce all three opsins. But as you go down deeper and deeper, so you get to the deep sea, there's a lot of species that live down deeper that lose the red opsin. They no longer produce it. Why? Because red doesn't penetrate much beyond about 30 feet, right? So red is the first wavelength to filter out. So why produce a protein that serves absolutely no purpose? Because there's no red wavelengths to detect, right? So in fact, we'll talk about this when we get into the deep sea, but one of the common things is many species of fishes or shrimps or whatever become red because red is the new black, right? In other words, there's no red wavelengths, so red is an okay color to produce, because there's no red wavelengths. And the colors we see is the wavelengths that are being reflected, right? So if there's no red wavelengths, there's no red to reflect. And so you can't see red. So it's as if it was black. Okay? Go down deeper, you get some species of fishes that basically only do blue. Okay? And then there's another one called rhodopsin. You produce this as well. When you get up in the middle of the night to go to the bathroom or the midnight snack or something, it's pretty dark. Rhodopsins are the ones doing most of the work at that point in time. A lot of deep sea fishes will have blues and rhodopsins and nothing else. So it's color blindness, but that is from an evolutionary standpoint, right? You can't see red, but it doesn't matter because red doesn't exist, right? If red did not exist in our world, we probably wouldn't have rhodopsins, right? And even though it does exist in our world, not all species of mammals, for example, can see red. So deer can't see red. That's why hunters wear safety orange, because deer can't see it, right? But turkey hunter, if they wear safety orange, turkey's going to spot them a mile away because they can't see it. All right, anything else? Any other tangents? All right, last thing is migration, diadramy, and shoaling. This is where we figure out the difference between the shoal and the school.
The shoal are the ones we eat with the big nets.
Do what?
The shoals are the ones we eat with those big nets.
Mm-mm.
The school is everything
Mm-mm.
else. It should
All right, but we will then learn the difference. We will correct that degree of misinformation.
make me the
At least for everybody. I don't know whether it'll correct it for you, but I'll give you the information. Do with it as you will. All right, so migration is basically long-term movements for ecological purposes, such as feeding or spawning, right? So a lot of fishes, you might have a particular species of fish, let's say on a coral reef or something, that during almost its entire lifetime maybe doesn't move much beyond the area of 4 or 6 of these tables, or maybe the room as a whole, right? They don't move very far. Or maybe it's just up and down the hallway, right, for a bigger fish. But they don't cover a lot of area. That's not migration, right? Migration would be something like salmon that are going way up the rivers, right? So they're going way, way away for spawning purposes, right? Or tuna, because of their high energy demand, they cover a lot of area to find the food, right? So they will move very large distances at regular rates in search of food, okay? So when we're talking about migration, that's what we're thinking about. Now diadramy, dia meaning two, right? So this is basically moving between saltwater and freshwater at different stages of your life, okay? And there are 3 types of diadramy. So diadramy is the overall umbrella term to describe whether you're going from salt to fresh or fresh to salt. That's diadramy, okay? But you can go from freshwater to saltwater to spawn. So for example, that's what our freshwater eels do. So the freshwater eels that are present in our streams and rivers right around here, they spend most of their time in the freshwater, okay? So I'm going to draw a slope. So this is our freshwater, right? So our freshwater runs downhill. Do I have blue by chance? I don't. So we have an algae bloom. So we got a pea green ocean out here, okay? So eels, freshwater runs down, right? Everything runs downhill to get to the ocean, okay? Cata, if you use cata as the prefix, means down, okay? So catadremy, basically going downhill, going down freshwater, out into the ocean to spawn, okay? So catadromous fishes begin in freshwater, and for that freshwater to get out into the ocean, it has to go downslope, right? So freshwater eels, those here, those over in Europe, all go out into the middle of the Atlantic Ocean and they spawn. And then after they transform, they'll hang out as larval individuals out in the oceans for a while, and then they go back in the freshwater where they spend the rest of their life, okay? The opposite of that is anadremy. So cata means going down, Ana, in one definition, means going up. Okay? So anadromous, they're spending most of their time out in the ocean, but then they go upslope, go up freshwater to spawn, right? And of course, in the case of salmons, the fishes, the adults die, young hang out there for a while and would work their way out to the sea. Okay? You good on the difference between those two? All right, now you have some species that will switch between fresh and salt water, but not for spawning purposes, right? So that's amphidrony, right? So like amphibian is both, right? Sort of freshwater or water and land, but in this case, they're going back and forth between freshwater and saltwater, but not for spawning purposes, okay? So the one I'm going to use for this is Lintipes. This is a species of goby that lives in these high mountain streams of Hawaii. Okay, so they spawn in these high mountain streams of Hawaii, and then the larvae go tumbling down river and end up out in the oceans, and that's where they develop, right? So spawning occurred in freshwater, but the larvae develop out in the oceans, but then after their larval stage, they have to get back up into the fresh water where they're going to spend their life, right? So they're not making this switch for spawning, because spawning took place where they live as adults, okay? I'm going to play a video to tell you the rest of this story, because it's a really cool story.
Young Hawaiian gobies leave the sea behind to take advantage of these upland rivers. To get to the pristine rivers above, these tiny fish face enormous hurdles.
The water whooshing.
I'd repel that.
Above, there are fewer predators
Such a
and real
Jurassic Park
competition for food. So this plucky little fish begins her seemingly impossible ascent. Just days ago, her body transformed.
From the larval
Her
stage.
mouth moved 90 degrees downwards, so she could use it like a suction cup. She alternates between her mouth and another sucker made from her fins on her underside.
That would be the pelvic fins, right? Fused together like a suction
Each
dish.
one's body upwards.
I don't think the music's particularly dramatic.
And she's not alone.
The highest one that I'm pretty sure I remember this correctly, the highest known waterfall that they have successfully climbed is 300 feet.
Around 99%.
There's a few different species in this genus, and there's two different genera that actually do this behavior.
So, do they kind of experience growing pains? Like, I mean, like, you know, everybody kind of has pain sensory, but like, humans can sometimes experience growing pain.
No idea.
As I said, I wonder if the fish do that when their mouth is moving downwards in the larval stage.
I'm gonna guess no. I don't know, but I'm gonna guess no. Because flounder do something similar, right? So flounders begin like this. They swim upright, and then they trip, and their one eye moves from one side to the other. But because that it's a whole reconfiguration of bones, I'm guessing that their nervous system is altered. It has to change in some way along the circulatory system. I'm guessing it sort of shuts down on things. So I don't know that. But yeah, that's kind of a cool thing is the fishes can make that big old climb like that.
Okay, I was gonna ask why in the world they developed to do that, but it's because there's like less predators.
Yeah, it's the
That's
predation thing, right?
crazy.
Yep. So again, you can always say, well, why did that evolve? And it's just like it just did, right? It's the combination of mutations that occur, selective pressures, and so on. But yeah, there are far fewer predators there than there is even at the base of the fall, right? Just because most Bird predators and so on, no problem, right? But in terms of fish predators or other predators that would be in the water, most of them, they cannot get up there, right? So once you're up there, if you make it, if you're that lucky 1% that makes it up there, then you've got a pretty decent chance of living a much longer life. And
I just can't believe that was the best option, you know?
it's,
crazy.
It's not necessarily the best option, but it's an option that was selected for them.
So do some of the adult gobies still live at the bottom, or is it?
Not that I'm aware of. No, all of them are found high up. Anything else? Well, yeah, good.
I was this big. That hurt.
All right, so shoal versus schools. So shoals are an aggregation. A shoal is an aggregation of individuals. They could be same species, could be different species, but it's an aggregation that they are unorganized, right? So they're basically just sitting there and they're facing in different directions. They're just hanging there. Okay, and that's all a shoal is, basically just an aggregation of fishes. Okay, so you might see, so if you look, again, like on a coral reef, shallow enough where you've got some of the movement of water from waves, right? And it basically moves the fish back and forth. And you'll see this large aggregation of fish, and they just sort of going back and forth, they're just hanging out, but they just move back and forth from the movement of the water from the waves, right? But they're all just hanging around. I'm going to do this for the whole rest of the day. But that's a shoal, okay? But once they become polarized, meaning they're all facing and moving in the same direction, now you have a school. So the bait ball, right, where you got that or something where you're getting them, they're trying to avoid predation or so on, they're all moving in that big swirling thing, that's a school because they're now polarized, okay? They're moving in a particular direction. So one of the things that you can think about is that to some extent, a school is a type of shoal, but a shoal is not a school, okay? So one of the reasons that you end up having schools is one, when you've got a large group of fishes, that large mass makes it more efficient from an energetic standpoint, makes it more energy efficient for moving through the water. Just like where you got geese that fly in the V, right? And that first goose starts to separate the air mass, which makes it easier for the other individuals. When you've got a group of individuals, the ones near the front are sort of starting to separate out, and all the other fishes behind can draft a little bit, and it makes it easier for them. No single individual stays up front all the time, right? They're always sort of moving around, sort of share the wealth kind of thing, but it makes it more easier to move through the water, right? Another hypothesis for schools is foraging efficiency. It's basically a whole bunch of eyes that are looking for food. So when you have that big school, whether it's tunas or little bait fishes like this, they're out there searching for food. And so you've got a whole bunch of eyes. Once one or two individuals see something, they branch off to go get it. Everybody else follows along, right? Hopefully you're the one individual that gets there first, right? Because then you can have something to eat. But the idea is you got a whole bunch of eyes to find food, which increases the chance that more of the individuals in the group have something to eat. Then there is also the predator avoidance strategy. So the whole bait balls with the swirling thing. If you're on the outside, it's a safety in numbers thing, right? In a big group, there's a less likelihood that you as an individual get captured by the predator, right? And certainly as this is moving around, sometimes you're on the outside, sometimes you're on the inside. Individuals on the outside are more likely to get captured. But you might say sort of roughly equal probability that any one individual on the outside gets captured. So safety in numbers type of thing. And then also you can have schooling. We saw an example of this before, but basically where you come in for reproductive purposes, right? So you can have the spawning aggregations. Good on that? Luke?
Yep.
We happy now?